So, good evening. I welcome you all in this Science Dialogue series. As you all know, this is free Science Dialogue series, and number is more than 110. We have organized. Purpose is to increase overall knowledge pool in bioscience stream and the stream which helps the bioscience, not strictly biology, but the stream which helps the biology partially, not completely. All streams are required, all scientific streams are required to understand the biology, but mostly the related topic which will help you to understand biology. So, in this series, as you all are aware of about this synchrotron, use of synchrotron in life science, today's topic. This is new topic. This is I have also heard first time and learned briefly about this topic from today's speaker, Dr. Isha Raj, and she is PhD from JNU School of Life Science, and then she did postdoc from बहुत सारे कंप्लीट जो है इंस्टीट्यूट के नाम जो है वो, but she did postdoc and she is recently in Sweden and she's coordinating the education and training workshop etc in Sweden. She's a prestigious institute. She may also introduce during her lecture in briefly, but see, we should utilize her knowledge. She has done during PhD. She did work on crystallography. So, as you know, crystallography required different knowledge of different domains. So from here, she went for postdoc. Several years she did postdoc, uh, and then she's in Sweden nowadays. We will utilize. She has also published several research and review paper, more than 15 research paper. So we will utilize her and learn from her this synchrotron new topic for us, not for all, new topic which is utilized in life science subject. So I welcome her in the Science Dialogue series, and I'm grateful to her that she is ready to deliver lecture under this banner of Science Dialogue series. I request you all that you can interact with her, Anna, during, if you are finding any difficulty during lecture, you can unmute and speak. She will reply you, Anna. So with this brief note, I have not read a long introduction. Uh, with this note, I welcome to Dr. Isha. Now,
Thank you, Gopal Ji.
Madam,
Thank you, Gopal Ji, for
yes,
that nice introduction and for this initiative. I really like interacting with people who are with, especially students who are up and coming, because they have a lot of ideas and they are the future generation. Especially in India, we have a lot of ideas that can be translated into really good research. The problem might be awareness. So I'm very happy to be here today to share knowledge about one of these fields. And if I
and
can figure out
participants are
how,
research scholar as well as a student, sometimes faculty also.
yeah. Don't
Yes.
worry. Yeah. So yeah, I would like, like Gopalji said, you are free to ask questions in the middle of the presentation, but I would also have some time at the end of the presentation for you to ask questions. So if I can figure out how to share the slides again, I'll do that.
पहले शेयर करिए
Mhm.
entire screen and
Mhm.
then they will ask the presentation share.
Yeah. Do you see the screen?
Yes,
Yes, I
yes,
think
yes.
so. Yeah.
Full screen.
Uh-huh. Okay.
Yes, yes, fine now.
Okay, so I'll start now. So what you see in the background is a synchrotron. It's like 520 meters ring. It's almost, yeah, it can be long, it can be bigger, it can be like 1 kilometer or whatever. It's basically a place where you harvest the light generated from electrons moving in a circle. And wherever it is bending, you harvest the light and use it basically as a big microscope for x-rays, and then you do experiments on different materials. Yeah, so today what I'll be talking about is how do we use the synchrotron light for life sciences. Uh, the outline is briefly introduction, synchrotron. Of course, the big question: what is it? What are the experimental methods that are used at synchrotron for life sciences? Some examples, and of course, if you want to use it, how do you go about finding access? So, a brief introduction about the electromagnetic spectrum. Uh, light or electromagnetic spectrum can range from a really, really big wavelengths where they are radio waves, and then you can look at the buildings. That is how you detect how submarines are in the water. And yeah, radio waves can also tell you where humans are located. Radar and other things, you use how that to detect those things. And then you have microwave, and yeah, microwave, infrared, and the visible wavelengths, where you can actually see that allows us to see things, because the wavelength is in the range of things that the wave can interact with. So the visible light interacts with things which I think you can just see up to protozoans if they are big enough. And if you use a microscope, you can see that through visible light. But as soon as you start going into ultraviolet and x-rays, normal microscopes are not enough. And x-rays, for sure, you cannot use normal microscopes to use that. So there is where a synchrotron comes in. So the light generated from the synchrotrons can uh, is in the range from infrared, a little bit of microwaves, to x-rays. So what we can harvest, it depends on the lenses or the optics that we put in place. How do we use it for in life sciences? I'm taking an example from max 4. So we have a number of beam lines. I'll shortly go into what beam lines are. But for example, if you make your lenses such that you can use X-rays in the softer, when I say softer X-rays, it means not very high intensity, not which penetrates the atoms or something. So it can look into your organs, but only at a certain depth. So those are soft X-rays, where we will use mid-max, which we still have not got funding for, but you can use it to see animals, organs, tissues. We already have Nanomax and Softimax working in the soft X-ray range, and there you can see cells and microstructures, like how the nerves in the brain are organized. And then you have the more traditional way of life sciences, which is COSAXS, Biomax, and Micromax. So they use X-ray scattering and X-ray scattering, X-ray absorption. Mostly COSAXS, Biomax, and Micromax, they use X-ray scattering to look at how atoms are organized in your protein. So we see that X-ray synchrotrons can be used for all these studies, but then what is a synchrotron? That's the big question. I'll try to briefly go into what a synchrotron is. So we usually have an electron gun here, where, if you can see my mouse where I'm pointing, we usually have an electron gun here, and then we use electric fields to accelerate the electrons to almost the speed of light. Of course, electrons have a little bit of
पॉइंटर
width.
यूज कर लीजिए वहां, उसी में एक पॉइंटर का ऑप्शन होगा ना, लेजर पॉइंटर, उसमें आप राइट में, हाँ
uh,
वहां पर हम देखेंगे होगा कहीं लेजर पॉइंटर।
Visible? Yeah? No.
हाँ,
It's- uh,
यस,
uh,
रेड
do you
भी
see the
होता है।
pointer? I don't think you.
हाँ, ग्रीन दिख रहा है अभी ना,
Mm-hmm.
रेड।
The gray green. Sorry. So can you see it now,
हाँ
or
हाँ,
do you
नहीं
see
अभी भी
now?
वही है, वो नहीं आया, पॉइंटर नहीं आया, कर्सर दिख रहा है वो, है ना, इसमें एक रेड आता, लेजर पॉइंटर कुछ कहीं ऑप्शन होगा।
I don't see that.
अच्छा,
Okay, I'll just
लाइट,
still try.
ओके, हाँ हाँ, ठीक है, ठीक, प्लीज कंटिन्यू,
Mm-hmm. I'll go for the automatic one. Okay. So you have an
प्लीज कंटिन्यू।
electron gun at the end here, and then the electrons are excited. I am looking at the rightmost side of the. I'm starting from the rightmost side of the image. So you have an electron gun here. We heat a tungsten, usually a tungsten metal, to very high heat, and then we use electric fields to get them to accelerate them in an electric field. And we also use radio waves to do that, which is called, usually it can be a linear accelerator, it can be another booster ring. And then you accelerate electrons to almost the speed of light. And once it has achieved the speed of light and you have decided what energy you want to work with, you let the electrons run in a storage ring. This polygon is called the storage ring, at a constant voltage. So it's a really high voltage. It's in the order of depending on what use you want to have, it can be from 1.5 giga electron volts to 5 giga electron volts. There's one which I think is even higher. So the more electron volts, the more energy the electrons can have, and you can do different kind of experiments with that. But for most biological purposes, 3 giga electron volts is more than enough. So what happens is, once the electrons are in the ring, you because you want the electrons to move in a ring, you bend them. And if the electrons are bent at the speed of light, they release is light. Actually, this was light or radiations, which was considered a nuisance in the beginning when particle accelerators will build, because then you have to make sure that everything should be safe. But and you had no use with that radiation. But some clever people figured out that that radiation can, if you can tune it, you can use it for different kind of experiments, because it is also a valuable resource. So we have two types of bending magnets. Wherever there is a bending magnet, there will be a radiation coming out, and this radiation you can use for your experiments. There's another kind of a bending magnet, which is called an insertion device. I will not go into the detail of what is the difference between them, but for now, it's like you will have radiation wherever you are bending the electrons. And then you can build a beam line where you can use this radiation to do your experiments. We can understand it a little bit better if we look at this animation. So there's an electron gun, there's a linear accelerator, and then it goes into the booster ring, and then they are transferred to all these into the storage ring, and then to these beam lines. These round spots are actually where the beam lines are. A better representation is something like this. So this round is what a beam line is. So first you have these optical part where you select what wavelength of light or what wavelength of radiation you want to work with. And then you send it to the experimental hutch, where this red thing is where you place your sample. And then you have a detector. And then you have a working station where you can control, like you do with a microscope. You adjust the focus, you adjust like where you can place your sample. Yeah, those kind of things you can do sitting in your control room. And then you can analyze the data once it comes out of it. So there are actually many synchrotron facilities around the world, almost 60. And we have actually one in India. It's called Indus 1 and Indus 2. So they had an upgrade of Indus 1, which is called Indus 2. I'm not sure if they are doing life sciences, but for sure they are doing material science experiments at Indus. And this is in Indore. But yeah, across the world we
किस
have
इंस्टीट्यूट में है? Where it is?
uh, in Indore
IIT?
it is नहीं नहीं
IIT?
राजा रमन्ना नहीं राजा रमन्ना Center for Advanced Technologies is called
Okay.
that. RRCAT. In Indore. It's a I'm not sure if they are using life sensors, but yeah, they will. I saw something about imaging, but yeah, they will. Uh, yeah, if there are a lot of people interested, maybe they will consider to have a beamline for life sciences as well. But anyway. So, what are the experimental methods which are basically used at synchrotrons? I am just broadly categorizing, and then you will have different under, for example, imaging. There are also different levels and different types, but broadly, we have three different experimental methods. which can fall into these three broad categories. One is imaging, where you display, if you are imaging something, it's like taking an image of a hand. So it can yeah, but it's also in 2D and 3D, so you can basically see how something is organized in 2 and 3 dimensions. Then there's a spectroscopy, where you understand what are the elements contained in a sample. Is there potassium? Is there calcium? What is there in the sample? So basically, you use light to interact with the atoms in the sample, and then take the readout and see, okay, because different elements interact differently, or absorb or emit fluoresce differently when light is shined onto it. And then the third, and which I had used mostly, was scattering and diffraction, where you put a protein crystal in the way of the X-ray, in the path of the X-ray, and get a scattering pattern, and then back calculate how it looks like. But of course, it's true for protein and also DNA. You can do a lot of a lot of structures have been determined that way. So just a brief overview of how those experiments look like. I'll go with first with the scattering diffraction experiment. Here's the X-ray coming. Here you have your sample in the path, and then you get, according to the sample, how atoms are organized in it. You get these diffraction patterns, and then this diffraction pattern is collected on a detector. This detector is connected to computers now, and then yeah, you can use this data to back calculate to determine how the atoms are placed in your sample. So that's a scattering or diffraction experiment. Um, then you have the spectroscopy experiment. Uh, Light comes and then it is modified by the sample, and then you take an absorption spectra and see, okay, what does this spectra tell me about the sample's electronic state or chemical bonds. And then I think the most easy to understand is the imaging experiment. So X-rays penetrate material in varying degrees, and depending on you can always tune your X-rays to see to to go as deep in the sample as you like, and then you can see varying levels of structure. We'll see an example of this later on. So that's imaging. I'll go with the first example, which is X-ray, which is X-ray scattering. And there was one example was that was done at Max 4 actually, was determination of an enzyme structure which could modify the B antigen on our red blood cells to see if mhm. Now, can somebody tell me? So we have this A positive, B positive, and A positive, B positive, and this negative O negative and AB blood groups. So what happens is O negative is the universal blood donor we know. So what they did is they found an enzyme which is an exoglycosidase, which can take away all the antigen on your blood cells, and then you can actually generate a universal blood for donation to anybody, so which was very cool. So this the structure of the enzyme was determined here at Biomax, at Max 4, and also at Petra 3. So there are complementary techniques at other synchrotrons. So yeah, this was done here. Then there was also what you can do is also look at a reaction occurring in real time. So you take your protein sample, you add your ligand to it, you pass it through this capillary, for example, and then here the example is you activate the reaction by a laser pulse, because we are working with an NPE caged ATP, yeah, which is nitrophenylethyl ester. And when a laser pulse is passed, or laser pulse is illuminated onto the sample, this ATP is then released, and then you can see how adenylate kinase is interacting with ATP in real time. And you take a picture on the detector, and you see these white arrows and the blue and the green structure. So that is how the protein has moved in the presence of ATP. So you get real-time data about how a reaction is progressing. So those were some hard X-ray examples. But then you can also do a lot of soft X-ray imaging. For example, you have this is used in case the example that I have taken here is how a nanoparticle, if you want to target certain kind of cells, and you are using a nanoparticle to target the cells, so what can you do about that? So you can decide to do hard X-ray imaging, or you could do a soft X-ray imaging. You do hard X-ray imaging to see how thick the cells are, where exactly the nanoparticles are located, and how is it interacting with the cellular architecture. And you do the soft X-ray imaging to see just the distribution of how the particles are distributed, and see in contrast, like at what level are they accumulating the most. So you might have thought that the nanoparticle will target certain things, but maybe that's not true. And the way it is interacting with different cellular organelles is different. So now what you can do through soft and hard X-ray imaging is decide how that is working. But maybe this is a bit, yeah, I will just skip this one. I'll take another example. This is, for example, this is this was an experiment done at Nanomax. What they wanted to see is when you have a fracture, how the fracture starts healing. So they took some mouse, and in this experiment they induced a fracture in the mouse, and then they monitored over time what is happening at the fracture site, how the fracture is healing. So they could see what are the elements that get deposited in the fracture once it starts healing. And you can see from X-ray fluorescence and wide angle X-ray spectroscopy that calcium and zinc and iron and zinc are the most common, how they are localized and where they get deposited exactly in the bone. So that also gives you insight into about nutrition, and also the process of healing. Like you can think about what is the best way, that or how can you speed up healing. Then not only animals, so you want to study plants. Actually, you can also do study biofilms, how biofilms are forming through soft X-rays. One of the example is at Nanomax is how do you look at a plant pollen, and again, which is the richest area for where zinc gets deposited or calcium gets deposited, and so you can image it at Nanomax, which is a combination of both imaging and spectroscopy. So the imaging gives you the shape of the pollen that you can see, and spectroscopy tells you that in this pollen grain where which element is deposited. The same similar example is also for Softimax. So what happens is a few of the beam lines are specialized for, not all beam lines can detect a lot of all elements. Some, for example, potassium or iron is a little bit difficult to detect, and then we have Softimax to do that. And then, yeah, here you can see they have looked at the endosperm of wheat, and you can see where iron, zinc, and potassium are deposited. And you can also, it's a not you don't label it or anything, so you are not you can actually expose your sample here, and you can let it grow. For these kind of experiments, you can also they don't get destroyed, so you can continue your experiment after that. Having said that, the mouse experiment was a bit harder X-rays, so yeah, there you have to sacrifice the mouse, but this can be used again. Then there's a very cool new technique which we are not doing at Max 4 yet. We still need funding for that, and we are this has been developed at actually at ESRF. ESRF is in France. I forgot to write the name, but yeah. So this is yeah, and this technique was developed with funding from Mark Zuckerberg Foundation. So what happens is uh, you you can image the whole lung, as you can see here, and then you can go much you can go to different levels. So this is the most highest level where you can see the lung. This is from a dead COVID patient, and then they looked at different levels. So this is a 2 micrometer local scan and then a 6 micrometer local scan. So it can characterize the damage in a lung lobe, which you cannot see with a normal CT scan. A normal CT scan will not give you this many details. So what they can find is uh, um, using AI. If you have so many details here, what you can do is you can use AI to connect this very detailed data from hierarchical phase contrast tomography and link it with normal CT. So then you can say already what kind of damage is done uh, using a normal CT as well. And I have a small. This is again not done at my max force, but what I wanted to show you was also um, as a tomography, where you can see this is microtomography. So you can see that they used a real fly in real time because the X-ray pulses are so fast. So this is just done in 3 seconds. All of this is done in 3 seconds, and you can see how the muscles are moving in real time. And this is at one level, and then as a CT goes, you can take cross sections artificially through your you can tell your computer to, like, yeah, take look at the different cross sections, and then you see how the muscles are organized at different levels. You can look into the details of actually how the fly is managing to, which muscles is it using to fly. And this is only possible with the high X-ray intensity that we get and the pulse that we have, the pulses that we can put on the in this small time on such a tiny fly. And this is all done in situ. So the fly, yeah, of course, there will we cannot do this with living human beings yet, but we don't care if the fly gets a few mutations. So, but we wanted to see it in real time. So yeah, and then this gives us more information about how the muscles are actually moving, which muscles are moving, and maybe in future design some vehicles or some machines which use the similar technique to maximize energy usage. So yeah, so yeah, I talked about a lot of, I hope, cool things what you can do at synchrotrons. But what do you do if you wanted to? Okay, you think that maybe you are doing an experiment which could benefit from using synchrotrons. In general, most of the synchrotrons are free for use to public because it is funded by public funding. So it is free to be used by the academic population, as we say. So the only thing they require is they are disseminated in open access journals. So what you do is you write a proposal, and all of these beamlines or all of these synchrotrons have calls for proposals. You write a proposal and you submit it to the synchrotron and see if they select it. The chances that your proposal will be selected becomes better if you have already talked to the beamline scientists, because they know that, okay, this experiment looks like that you have an idea of what you're talking about, what experiment you are going to do, and if the beamline has the capacity to do that experiment. So the chance for a proposal to get accepted is increased if you have already talked to either a beamline scientist or a person who is knowledgeable about synchrotron techniques and who can guide you about how to write a proposal about, which is good at experimental guidelines, and which will pass through all those checks. Most of the beamlines have usually 2 cycles of calls, um, but I'm not sure. Some can be open throughout the year. You can submit a proposal anytime, but I think most of the synchrotrons will always shut down for 1 month or 2 months, so that they can do all the maintenance work. So that's why the calls for proposals are divided in spring and fall. What I mean by, at least at Max 4, when it's a spring call for proposal, then you get to do your experiments in fall. And if it is a fall call for proposal, you get to do your experiments in spring. So yeah, yeah, if you want to know how to apply and about those, you can use the QR code here. Yeah, Maxport does not provide travel and stay arrangements. I know that in the past, India had these collaborations with the France Synchrotron, through which I came for the first time to France to collect my data on a protein crystal. And with Germany, we had a different project where I could also go to Petra in Germany. So yeah, there are different projects which allow for international collaboration and through which you can do the travel. Yeah, but having said that, Maxford does not have any such collaboration right now with India, so their travel and stay arrangements are a user's responsibility. But yeah, if you have an idea for a project and you think you can manage the funding for your travel and stay, you can always talk to a beamline scientist before submitting a proposal. And it's true for, I think, anywhere in the world. It's not only true for Maxford, everywhere. Um, thank you. And if you have any questions, I would be very happy
Thank
to
you. Thank you.
take.